quality control function is one of the critical functions in the pharmaceutical industry. The quality control function is responsible to ensure that the drug produced meets SISPQ, that is, safety, identity, strength, purity, and quality before the life-saving medicine reaches out to patient. Recent trend of inspection observations reveals that the US FDA has increased scrutiny of this function, and non-compliances observed are also significantly high in this function. At Dr. Reddy's, quality control consists of about 1,700 personnel, internally managed into multiple sections like raw material, in process, finished and stability, etc. About 14,000 samples are tested and released every month. The QC laboratory exists in an environment that produces large amount of data. The paper-based execution process has its own inherent deficiencies, in addition to added complexity in handling paper records for its lifetime of at least 3 years.
As a team leader of this section, my responsibility was to ensure and improve SPA and TAT. In the paper-based process, it was becoming difficult with lack of visibility of pending, in progress and completed samples, inadequacy in monitoring workload under each analyst, transcription of results from ROA to SAP.
To perform the sample testing, in addition to performing actual testing, there are various tedious activities involved, such as waiting for the sample allocation and the collection of the record of analysis. And we need to ensure each and every resource, such as the standards, the solutions, chemicals, and the instrument we are using are within the validity period. And we need to print the report from the instruments and transcribe the raw data to the ROA. And we need to manually calculate the final test results and round it off as appropriate.
As a reviewer, I had to verify each recorded entry, its associated calculations, instrument reports, logbook entry for Alcoa Plus, which consumed a lot of time and efforts. There is also a possibility to miss the errors in the records which are generated.
Major constraints with paper-based execution are 1. Availability of resources for execution. 2. Incidents on good documentation practices, calculation errors. 3. Checklists with about 160 check items per batch. 4. Each data point review and multiple reviews resulting in increased cycle time. 5. Number of papers produced, 10 lakhs per month.
Quality and ITBP team at Dr. Reddy's embarked on a journey to transform quality control processes using digital technologies. To digitalize the core execution process, the LIMS program started with an aggressive target with an empowered core team. We targeted for minimum viable product that gives maximum benefit, that is samples from finished product. We adapted the digital lean principles for design and implementation of the application at all our sites. While implementation, we also focused on building site level capability to operate digitalized process in a sustainable manner.
The system captures data from source and it also has checks and balances for the entries and making into system. The controls available in the system enables lot of time and efforts were taken in the manual process. After digitization, we are able to perform the analysis with automated systems in less time and more accurately.
We utilize this opportunity to relook our end-to-end process and numerous simplifications were achieved. Major simplifications were reducing the process step from 10 to 4, chronographic process steps from 9 to 5, and eliminated at least 110 check item per batch. Apart from this, there were 21 processes were eliminated, 24 process automation were done, and 21 process interlocks were created. This implementation has helped us to simplify our operations.
Controls that are available in system like tolerance to the parameters, validated formulas, auto updation of resources, controlled validity periods, simplified the review process, and to review exceptions. System determines the complaints to predetermined specifications and upon completion of all approval workflow, generates the certificate of analysis automatically.
More than 100 sample sets in chromatography are reviewed on daily basis in FTO3. Across pharma industry, this review is currently manual and each sample set takes about 20 to 25 minutes. The prize winner idea from Exponential Day 2018 to automate this process using robotic process automation technology, that is RPA technology, was under implementation. RPA ensures review in consistent way by eliminating the possible human errors. Also, the review cycle time is expected to reduce significantly. More use cases are identified for automation using RPA with supervisory decision making by our analytical quality assurance team.
Digitalization of core operations opened up opportunity for continuous operational improvement using analytics. Descriptive analytics like turnaround time, out of specification and out of trend work team level efficiency monitoring and instrument utilization have enabled us to improve efficiencies in our operation on a continuous basis. A day in the life of each one involved in testing and release activities is getting transformed with SLA and metric-driven processes monitoring. The day starts with looking into pending numbers of samples for testing and release, SLA's adherence, compliance to quality notification requirements, pending action items, all enabled with a single version of digital truth through dashboards. Predictive analytics tools, which are created by applying suitable algorithms on huge amounts of data, became really helpful to take proactive actions. Some of those are drug product or drug substance stability prediction and prediction of chromatography column life. Processes those used to take several days to compile data, such as data trending, investigations, continuous process verification, are enabled with a click of a button.
There is a lot more that LIMS can do in terms of assuring the quality of the product to the external stakeholders more than even for ourselves. For ourselves, of course, we know our strengths in our analytical tools, analytical methods and various processes that we use. Whereas to the external world, when we tell them about LIMS and the data coming from there, which is not handled in a manual process at all, that gives a very special kind of capability.
All quality control operations that are executed and controlled through electronic systems will help us to maintain A. Good manufacturing and laboratory practices. B. Ensure data integrity. C. Facilitate continual improvement. D. And faster delivery of quality products. To address patient needs. Because we all believe good health can't wait.